over the course of the next few minutes, we're going to listen to some consortium briefings. But before I tell you what's going to happen with consortium briefings, I'd like to remind you a couple of things. You might have noticed the agenda kind of rolls into lunch. That means there's no formal get up and take coffee. Instead, take your time to do it when you please. There's coffee, tea, water, pastries, fruit, all distributed either through the end of the hallway or in the poster gallery. So take your time to do that at your own leisure. Not everybody at the same time, okay? Over the next few minutes, we're going to listen from our consortium leads. We are trying to raise some of the junior voices in our group. So you'll notice that not all in this slide are the usual suspects. From the grand challenges, we'll have Mark Christopher talking about AI-ready salutogenesis. Trey will share updates from CN4AI. Erich will share updates from Chorus, and Yael from Voice. On the working group side, we'll have Harry Caufield sharing with us information about standards. Lucila will share information about communications and meetings. Goldie Komaie will be sharing information about teaming and collaboration. Camille Nebeker will share information on the LC working group. Alex Buey will share data sharing, dissemination, and open house. And then finally, Jack will bring the rear with training, recruitment, and mentoring. So each of the grand challenges speakers, listen to me carefully, you have 10 minutes to share your presentation. And each of you working groups, you have 5 minutes each. We will time you. Julia Levites-Trecalova, who's sitting over here, will show you that big purple thing when you have 2 minutes and you need to hustle. We will get up and throw you off the stage. And then we will have a Q&A session for about 30 minutes for everybody to ask questions about all of these programs. Does that sound like a plan? Let's get started then. Oh, yeah. Mark, just kidding. We're going to get Erich to go first.
Next door, we have aim-ahead trainees, and they're going to be presenting to us their work. So I wanted to make sure to come there, and then I'll come back for questions if it's okay. So it's a lot to follow that act, and thank you to everybody being here. I just was reflecting as you're introducing the program and hearing from Vivian and Haluk that I remember talking to one of the preeminent coma scientists in the world, and he was saying, what you guys are doing, Bridge2AI, is really interesting. If it actually works, there might be a lot of data there, and it's nice to be able to stand up here and sort of talk about what that means when it comes together and we're all here and the program is successful and not just a bridge center or one project, but so many different projects all together. So I'm going to present on our program. This is called a patient-focused chorus, or a collaborative hospital repository uniting standards. And this is the clinical care AI Grand Challenge. And so I'm presenting on behalf of many of our investigators. And the disclosures we'll present to you is the funding for this program that we're here talking about, the Aim Ahead and Bridge2AI collaboration, which is in the room next door that I mentioned. And then also we received some grant funding from Microsoft's AI for Good program, which helps us host our data and perform compute as sort of cost sharing. There's, for real world data, which is a unique topic for our program, there is a lack of AI ready data available for people who want to develop foundation models, do comparative health, develop digital twins, and so forth. If you want to validate a model and send it to the FDA, typically you need data from more than one hospital to show the robustness of your model. And so in our program, our goal is to develop a multimodal, multi-center, and high-resolution data set of real-world patient data for over 100,000 admissions. And so we are splitting that into 60% for training that anybody should have the ability to work on, 20% for testing and data challenges, so that no one can cheat the test. We know how the results are on a data set that they haven't trained on, and then 20% to even take a harder test, which is one if you want to apply for regulatory approval. So many different investigators in this room. I hope you'll meet some of them at the many posters we'll cite. We're from around the country, and our goal is to take on the bottom these different modalities of data and standardize them in a way where they can come together in a unified capacity. That means at 14 different hospitals, everyone has to do the same thing, which it turns out is not a very easy thing to do. No surprise. We're all, quite different. But on the bottom, we're standardizing electronic health record data to OMOP. This includes flow sheet data, medication administrations that are barcode scanned at the time that the medications or infusions are given or adjusted, notes, labs, diagnoses, procedures that are performed, imaging data from DICOM in there, as well as cardiac telemetry. Those waveforms and images are being standardized adjacent to OMOP with an extension that's been sort of custom built through work that's been a collaboration from the Odyssey team. We have members here who've really been leaders trailblazing these extensions, and there's a poster I'll talk about, so that data can be searched in OMOP as well. And then geospatial exposomal data, which also has a GIS extension. We have an expert in the room who's worked on that as well. And then metadata about each hospital, so you kind of can normalize a patient to what the treatment is at that hospital. I won't go into it. This is an example of a waveform extension. Brian Gao, sitting in the front row, has a poster I hope you'll walk by. And Jared Hoteling and others have really been trying to help bring waveforms, which are objects, they're files, into OMOP, which is a table. And so that's not an easy thing to do, but representing the metadata and what you can search for to understand that, is there an ECG lead 2 on a patient where, heart rate variability has been computed in this way. You'd like to be able to search that and find it. We also have Jackie Lee in the front row, who's been working with Brian also and others at University of Toronto through a collaboration between our group and the group at University of Toronto to really understand what happens when you get real world data and it has all these artifacts of being acquired in the real world. And what happens that we're finding is that there are gaps and time warps and shifts. And I hope you'll stop by Jackie's poster and you can see on the bottom left that sort of clustering of where these weird warps and gaps in data occur. And the punchline on the bottom right is that if you start computing metrics like heart rate variability, you'll find there's huge differences in the metrics that you compute on the raw data versus on a repaired data. So as we go repairing real world data, we're sort of repairing our world in a way. But when you do this, that means when you run this algorithm in real time, you want to be able to do something like this in real time, so you get the right answer. So you don't have a 47.3% difference in your PNN50 because you did it on the raw data rather than repaired data. So in our project where we're trying to collect 100,000 patient samples, we are 90% of the way there. That's an incredible achievement to think about what I said in the beginning about, if this project succeeds. The data now has 400 active users in a trusted research environment. We'll talk about sort of the ethics of why we've chosen that strategy. We estimate, we're prorating now, that we're at about 3 billion rows of OMOP EHR data in that 90,000 admissions. So, if a nurse documents the pupil function at 9 a.m., that's a row. But just gives you a sense of the magnitude. We estimate that the waveform data in this 90,000 will accrue in the hundreds of terabytes, and that we're starting to de-identify our imaging data, and we estimate somewhere above 2.5 million studies. Some of the largest public repositories are in the range of 200,000. We completed with the standards working group a really nice a publication of our metadata using an RO-crate method that came through one of the other DGPs through the standards working group. It's just a really good example of cross-bridge collaboration. So now our metadata is up. And we have a host and a suite of tools. I don't have the time to show you them. One of them is just a place to run data challenges in our research environment. We've run about 5 or 6 of these. Another coming out of Emory is a tool for agentic workflow where Tony Pan and some of his team are here. There's a poster on that. We hope you'll stop by. But this is from Del Bold, also at Emory, where you say want to type in, I'm I'm your research assistant, or you'll get that and you'll say, well, tell me about type 2 diabetes. And it says, I found 1,240 patients matching the criteria for type 2 diabetes. And here are sort of the distributions of that and so forth. So we're sort of trying to, by the end of this year, get to a point where people using either low or no code can help with some of their data wrangling, which is really one of the, probably the main component of any time people spend in data science. And similarly, there are different sort of panels for our integrated viewer. If you wanted to say, choose a patient, pick it and pull it up. And now you're searching here for pain. I'll speed through some of these because Moni, I want all my 10 minutes. You can pull that patient up and sort of see where they are. The work I mentioned from Tony through the Georgia CTSA is a nice poster on that as well. Is now trying to through Emory and the Georgia CTSA be able to use agentic queries. So here's a question: find all the patients diagnosed with melanoma. They've done this. And the work they've done here, they're trying to bring into the Chorus data set so that we can sort of use natural language to start querying our data and use exposomal data. So here you can see, clustering in Atlanta is a hotspot of melanoma. We want to see where the exposures are that, for example, might lead to trauma. I'll skip this slide, similar work. Morteza Zabihi, who can't be here today, developed a dashboard so we can sort of start translating data to presenting it to clinicians and families and interpret data. So if you predict recovery, what's on the bottom left, the stability of that prediction. On the right, if you had dials you could turn and say, what if my patient was 27 or 28, what would that do to a prediction? So this is all about interpretability. And then on the punchline, we've been asked about sustainability. We now have 2 proposals from some of the cloud service providers to sustain our data set hosting for the next 5 years. We're grateful for those, and we're sort of weighing those and how to engage with those groups. And we're now in industry discussions with about 6 or 7 industry groups who'd like to use our data to help people, which is really amazing. I would say from the sustainability standpoint, a real question for us, what I hope we'll get into in this meeting and Camille will allude to is, we are creating and sharing a representative real world data, clinical data set. And so it's important to understand that because this data was not consented, it's historical data from the medical record, the common rule does exempt secondary use of that data, but its use is regulated by HIPAA, the HIPAA privacy rule. And so access to that complete representative data is great because it does not have a consent bias. And it does allow us to share that data with HIPAA-covered entities for their use in secondary research and for use in public health. The challenge, though, is that we need to come up with a legal scholarship-informed design. 2 minutes? 1 minute? Okay, good. Which is how to share that data beyond a HIPAA-covered entity like a hospital. Okay, and so if the government takes ownership of that data or delivers it to someone else who is not a HIPAA-covered entity, some of the security protection or some of the privacy rules collapse. And we need to do what is called de-identify the data. So we either have to go back to all the patients and reconsent them. We can't find them, and some of them are deceased. We cannot do that. This slide sort of talks about that, sort of impractical. Or we can de-identify the data. There are 2 ways to do that. You can achieve safe harbor, which is any unique characteristic of a patient has to be removed. The problem is now we know, your Apple Watch tells you that your ECG is you. Your brainwaves tell you that you're you. The fact that you had porphyria in Texas in 2021 and you succumbed to sepsis tells you that it was you. And that is a very, very hard thing to do. And one of those true de-identifiers is that you can't re-identify the patient. So the next is expert determination. And either you get an expert to tell you that all 100,000 patients and all half a petabyte or petabyte of your data is de-identified, which is a very expensive thing to do. Or you can choose an expert determination method, which is that we as the experts put it in a place where we know no one can re-identify it. And that's what a trusted research environment is. You come into the enclave, you compute, and we audit you. So if we give you the data, you download, you take it home, we cannot audit you. You can sign a contract with a data use agreement, and we know that those are not really legally enforceable, and they are not on paper meaningful. And so that puts our hospitals at some jeopardy. And expert determination methods, we expertly determine that in a trusted research environment, you cannot re-identify our data because we audit you and you don't have the time to do it. So that's where we are. And so we're trying to figure out how to do that and yet open up this enclave and have the level and volume of compute to really accelerate AI. And so, we'll discuss, maybe Camille, you'll get into some of the other options. I'm getting the time check from Moni. And then the last thing is that, I'm sorry, but the government made it hard in December 2024 by challenging how we share data to countries of concern. And this not only means if I directly share, but if there's derivative sharing. So challenges for us. With that, we're doing our job. We're here. We look forward to answering your questions. I'm going to step aside to meet with the AIM Ahead folks and look forward to sort of hearing all your questions. So, thank you. There's lots of people.
Okay, wonderful. So other members of the chorus team will be here for the Q&A. That sounds good. Yeah, we'll bring you back when we need to. Yes. Okay, thank you. So Mark, is your turn.
Hi, everyone. I'm Mark Christopher from UC San Diego. Today I'll be giving a quick update on the AI-ready Salutogenesis project. So the AI-ready Salutogenesis dataset consists of data collected from people healthy and type 2 diabetes participants. And it's a dataset harmonized across 3 data collection sites. So the collection and the design of the dataset was geared towards use in future studies using AI and machine learning techniques. And so to this end, the recruitment and collection process and sampling procedures were aimed at achieving a distribution of both healthy participants and those across the spectrum of diabetes severity. And the collection protocols included the capture of a variety of different data types. These included survey data, physical measurements, clinical data, imaging data, and wearable device data. So a primary focus of this dataset is on salutogenesis. So that is a return to health as opposed to from healthy to disease. And that's in type 2 diabetes. So the subsets of this data are available at 2 different levels of sensitivity. So the first is a registered access data release. So that includes those data which are not considered sensitive personal health information. And it's available for download upon agreement with a license that dictates how the data can be used. So this data set includes survey data, blood lab results, fitness activity level, clinical measurements, retinal imaging, ECG, environmental sensing, blood sugar, and other data like this. The controlled access data level, this consists of more sensitive personal health information and can only be accessed by entering into a data use agreement. So this includes the zip code, sex, race, ethnicity, genetic data, health records, and medications. So as enrollment has been ongoing as part of the AI-ready project, we've released the data in an initial phase as well as yearly updates. So with respect to data collection, I'd just like to summarize what we've accomplished so far. So we have currently enrolled over 3,800 participants at 3 different collection sites. So this puts us on track to meet the total recruitment goal of 4,000 participants by later this summer. Based on participant surveys, they've also been happy with participation in this study, with 98% being happy or very happy with their participation and 96% saying that they would recommend it to others. So we've also engaged in community participant engagement and open house events. So over 200 past participants have so far joined, and we have another open house planned for later this year. In addition to collection, we also look at data dissemination. So with respect to the registered access data set, so this is the more publicly available data set, we just finished the year 3 release in November 2025. It consists of about 4 terabytes of data. And so far, for year 3 access, we've had 245 groups or individuals access this data, have approved access to this data. And overall, throughout the 3 years, we've had about 952. With respect to the controlled access data, we've had 22 data use agreements submitted, 20 of which have been approved, and so far about 10 have actually accessed the data. And again, finally, we also have engagement with users of this data, so not just disseminating data, but also helping people understand the data and use it more effectively. So that includes teaching courses at ARVO. So that includes ARVO, this upcoming ARVO in 2026, as well as data challenges hosted at the Komaie conference, the medical imaging conference this year as well. So I'd also like to talk about some of our teaming accomplishments here. So recently have finished the final teaming survey administered along with the Bridge2AI group. So this is a third survey since the project start and focuses on team dynamics. We also host in-person workshops to look at key milestones for the project and sort of facilitate collaboration and develop sort of strategies for our future work. We also spent a lot of time doing engagement with the Cheyenne River Sioux tribe. So this is part of establishing a spin-off project with direct funding from to engage with that community. And so far I've done interviews with both leaders within that community as well as researchers to get perspectives from both, and are currently collating initial findings. In terms of use of the data, so we have currently manuscripts and data analysis for the third manuscript, so identifying key engagement patterns of the use of the AI-ready data and trying to help us optimize the data set accessibility and utility. So in addition to data collection, curation, and dissemination, AI-ready also incorporates a skills and workforce development work, and that includes a year-long internship program. We are currently working with the third-year cohort, with the year 3 cohort, and that includes 6 interns from across the US. These interns have a variety of training levels, including current medical students, pre-doctoral scholars, and postdoctoral fellows. And they have a variety of focus areas that include ophthalmology, kinesiology, bioinformatics, and biostatistics. The internship program includes mentorship from both the AI-ready team as well as individual mentors for each intern. And this helps to guide professional growth. They provide guidance on specific individual intern research projects, as well as access to networking and collaboration opportunities, as well as just general career advice for these early career interns. The program focuses on, in addition to these mentorship opportunities, the program also focuses on engagement with the AI-ready dataset itself. So this includes developing research projects and using the AI-ready data for those projects. It also includes engagement by the interns in both the data collection and quality review process for the AI-ready dataset. Finally, the intern training includes didactic coursework, and that comes in the form of an in-person 2-week boot camp that we perform each summer. It also includes synchronous, and so that bootcamp focuses on AI and machine learning techniques, programming, data analysis skills, as well as exposure to the AI-ready data set and experience working with that. The training program also includes both synchronous and asynchronous lecture series, and of course, participation in events like this to give the interns experience presenting at conferences. So I just want to talk a little bit about the lecture series that we provide here. So the lecture series developed as part of the AI-ready training program covers a wide variety of topics that includes everything from the design of the specific AI-ready data set to more generally data standards, data formats, FAIR principles, and introduction to diabetes. It also covers technical topics like retinal imaging, imaging analysis, and machine learning topics. And finally, I'd also like to mention our collaboration with the AIM Ahead program. So the lecture and training materials developed as part of the AI-ready have been extremely valuable to the AIM Ahead training. And the current cohort of 25 AIM Ahead trainees have developed projects using the AI-ready data, and they are also presenting here at this conference today. So please check out their posters and their lightning talks. And with that, I'd just like to thank all the faculty, collaborators, the program and project managers, the developers, the clinical research coordinators, everyone involved, and encourage everyone to check out the AI-ready data set. And please come and see our intern and trainee talks. Thank you very much.
That was great. Thank you so much. I know all of the Aim Ahead trainees are very excited to be here, but I have to instruct you to go next door and join your leads for a check-in activity. So if you're still in this room, make sure you make your way to the next door over. Okay, so next up, our partners from the Precision Public Health. Voice. Sorry, that was the next number over. What's that? No, it's voice, yes. Brilliant.
Okay, I have a perfect this. And do you know where the video is there? But them told me it's uploaded.
Yes, once you're ready to present your video. It's right here.
Hey, hi everyone. There we go. So congratulations to everyone on all the data generation, but also the image generation that I could see with a lot of AI tools that have helped generate these PowerPoints for all the DGP. So congratulations. This is one example. Just to remind everybody about our DGP, we collect the largest flagship data set of ethically sourced voice recording for AI research across 5 sites. We have 15 sites in our consortiums, but 5 are doing data collection. And we'll talk about all the updates of this year. Really, from the last 4 years, I could tell you that disseminating voice data has been the most challenging. Collecting voice data is challenging. We knew that collecting voice data was challenging. Disseminating it is really interesting, and I could tell you a lot about what we've learned. So our major progress this year since the last F2F, in terms of our data, we did our year 3 data release, but the really big change this semester is that now our raw voice data is finally available. It took a long time. We had plans to release it really early. We got into lots of roadblocks due to privacy issues. The plans that we had in year 1 have definitely changed. We had to work a lot with the NIH, with all the different regulations that have changed. So it is finally available on Sage Bio Network, which has been our partner for dissemination. And I'll tell you about the updates about that. Lots of good data on the de-identification pipeline for free speech and the pediatric voices that we're going to talk about. Over 2,000 participants collected, and we finally got the breakthrough of remote data collection. We'll talk about that. In terms of data usability, we have our data usability task force. It's actually a group composed of clinicians, AI engineers, and students who see how we can use our data, and we have feedback loops to tell our people who collect data and who package data. And in terms of tools, we'll talk about the new Bridge2AI voice web platform. We're working on explainable AI methods. We have a poster about it. And that's it. So in terms of data collection, this is outdated because it's from last week, but we now have collected more than 2,000 participants, about 1,000 adult and 1,000 pediatric data. So this is just the pediatric data is the largest pediatric available data set in the world. Okay, so huge, huge, huge congratulations to obviously all our team, but our pediatric team is doing exceptionally well. This is some highlights from our tools that are available. So Alex Seguera's team, who's here from Cornell, now transitioned to a remote way to also collect data. So if you remember, we were collecting all the voices in clinical setting. It was really important to go from very high accuracy, validated, oh, there's Alex, and transition to possibility of remote collection. We knew that because we knew that a lot of projects were doing large, broad remote collection online for voice, and that they had to throw out about 70% of their data, right? So large data sets like the COVID data sets, the Coursera data set, a lot of their data was just not usable because people were doing it from home. So Bridge2AI Voice really worked on validating clinical data set, and now we're transitioning to home, and we're be able to capture the difference scientifically. This is an example of our remote data set platform that the TDOM team did. And then we also have internal dashboards where you could see the different types of diseases that we're collecting. So these clinicians usually that access the data, I love that dashboard because, for example, if they're working on Parkinson's, they want to know how many patients we have for Parkinson's, how can we get more. So that's been really exciting. This is our data releases. We started in year 1, and it was really important for us to release small amounts of data. And now in year 4, we're planning to release 3,000 patients. In our next data release, we're also preparing to release EHR data, which is very exciting, and the free speech data, which is the hardest thing to release. I don't know if you guys know why. So it's easy to, everything's hard, but it's been less challenging to release passages that are validated or what we call prolonged phonation like E, because there's a lot less PII involved. When we ask a patient, how are you doing today? That's called a free speech response. And there's a lot of PII that can come out of this. As we know, as we've listened to it, some people, even if you tell them, do not talk about your personal information, they say, I just came to visit Dr. Bensoussan and it is Monday morning and I live on, it's really interesting what people can say. So there's an incredible amount of work, both from an ethical perspective, from our ethics team, and from the technological perspective for Satra's team, who's here, to then use technology to be able to parse out the PII. So we're still working on that. We still have not released any free speech data because of that reason. I'm excited to report that we've had more than 400 downloads through PhysioNet. So that's really incredible. That's for our registered access. There's about half and half for pediatric and adult data. 400 groups around the world have downloaded our data set. We also have a controlled access data where we have our raw voice. So we protect raw voice to the highest standard. And we've already had 77 controlled access data requests. So that means that people say, we want access to your raw voice data. And then we tell them, okay, but you need to sign a lot of agreements for that. That's through a DUA with the university. And there's a rate of attrition that we're testing right now. At this point, we've had 13 completed packages that went through our DACO process. And we're trying to hold their hands to see how we can get them to complete the package. So we're very excited. We have not released any yet. So this is the process that's been happening. We continue to receive 2 requests per week, which is really huge. Our remote data collection finally got a breakthrough. Just to report, I love to report good news, but then we have to also be honest about our lessons learned. When we launched our data collection for remote collection efforts last year, we received more than 300 participants within 48 hours, and 99% were spam. Okay, so I mean, it's an important problem, right? When you ask the community to participate, we had people that just wanted the money. And then we had a lot of spammers, so we had to parse through that. So now we have a better system where we collaborate with foundations or patients group to do the remote data collection. This is a really fun research project on automatic gain control. Do I still have time? Okay, 3 minutes. So when you give your voice through a web app, okay, so if you give it through an app on your phone, the automatic gain control is great. When you go to a web app, what we noticed is that the computer analyzes any continuous amplitude as screaming or noise. So if you say E, then the web app will automatically input their automatic gain control and reduce the amplitude considerably. So that's a big problem for voice data analysis. So Shramana, who's here, our early career researcher, spearheaded that project. He tested all the different methods of different web browsers with different types of inputs, of plugins, to see which combination worked or didn't work. And so to give you an example, I don't know if it's going to share. I don't know if it's going to share. No, it's not. Okay, let's see. Okay. Well, you can't hear it, but you could see this is the signal if it was collected through an app. And then this is the signal being reduced if it's collected to the web app. So it gives you inaccurate data, okay? So we've had to work a lot this year on this problem. We found a solution and we'll present it at the next F2F. But it's a lot of work. Another really important problem, we've asked around to all the voice researchers, how do you do to proof your pipeline to remove PII? And they all said they have a great AI model that removes all the PII and PHI. I said, okay, have you released data? Oh, no, not yet, because it's not perfect, right? But we have the obligation to release the data. So we have the obligation to be 100% perfect and make sure that when we release, there's absolutely no PII and PHI for voice. So Evan, I don't know if he's here, another early career researcher, has listened to 300 files, 300 patient. So that's thousands of files, just to look at removal of PII and PHI and correlate with how the machine learning models are doing. So just so you know, the amount of work that's needed from an ethical perspective. We are happy to report that we are 100% compliant of the AI readiness score as of the Row Creates AI readiness score for our data. This is our different types of data set. I never believe anybody that says we're 100%, but I guess that's what the score gave us. So we're excited to say that. This is example from real feedback from people who email us about data access request. People say they're highly committed to the Bridge2AI mission, that we're excited about the opportunity to work with this valuable resource, highly relevant and widely used. So really great feedback from our teams. I'm done, almost. The funny one on the left is we often get spam to our DACO. So you could see here also we get spam from spammers that ask for a data request. Huge voice symposium this week. Next week, we're having a huge voice symposium. Bigger than the 4 years. We have 200 people attending, lots of people from industry. This is an AI-generated picture that says that it takes a village. We're very proud of our authorship, the Bridge2AI voice authorship. We spent a lot of time at the beginning of the consortium. We were very strict, and now we're happy to say that 24 papers have been released with the Bridge2AI voice authorship. More papers have been released from individual people in our consortium, and this is all the authors on our citations. We have some partnerships with industry. Voice Biome from Zana is going to be at our conference and provide their platform. We have collaborations with patient partner groups like the DBS Tempa, which is looking at depression and anxiety. And then this is some example about our data requests. So I think I talked about this already. Lots and lots of great papers from our ethics team and lots of great tools that they've already put out. So you can check these out on our website. In terms of TRM outreach, there has been over 5, I think, activities, hands-on activity workshops this year. So huge congratulations to our TRM teams. And we have a hackathon next week. We already have 50 people registered and 2 more workshops in clinical conferences. I love to say a little success story at the end. So these are our early career researchers and trainees presented at our national conference last week. And then Trevor Farr is one of the students who did a summer internship in the first year of Bridge2AI. And now he just got accepted and funded for a year of a research internship with us this year. And he now wants to be a voice researcher. So we're very excited. And the last great news is that we're excited to report that for our sustainability planning, we are opening a nonprofit called the International Society for Vocal Biomarkers. That's going to be announced next week. So you guys are the first one to hear about it. This international society will be run in collaboration with the Komaie Voice Group from Luxembourg. And our mission will be to host the yearly Voice AI Symposium, establish international standards in terms of voice sharing, ethics and regulation, promotion of our members' work, and advocacy to payer, regulators, and pharma. That's it.
Thank you, Yael. And last but not least, it's the CM4AI team. It's time for real, Trey, sorry.
Okay, thanks, Moni. I'm Trey Ideker at UC San Diego. I'm the contact PI of the Functional Genomics project, also known as Cell Maps for Artificial Intelligence. I'm going to go through a couple of slides to refresh people's memories, but I'll be quick. As I think most people in this audience now know, the main focus of our project, like many projects in functional genomics, is to greatly or seek to greatly enable machine learning on the human genome. And the way we're doing that here is by providing data and tools and methods to map the molecular pathways by which mutations and variants in the genome are translated to clinical outcomes. And the reason why this information is important, not just for machine learning, but for broad knowledge, is one, it does increase the power to translate the genome in terms of its predictions for clinical outcomes. Two, it is a great source of drug targets and points of intervention as you begin to map out and understand those molecular pathways. And three, perhaps most important, it's important for trust in the system. You don't want to have a system that's making predictions from your genome or any other data without that system being held accountable for the underlying reasons and rationale for its decisions. And that all really maps back to an understanding of molecular pathways in disease. The three major data sources which this project has generated and is continuing to generate are shown over here at left. I'll get into those in just a moment, but basically we generate three molecular modalities to reveal the structure and function of molecular pathways. In the middle here, we then integrate those using a series of tools to create derived data types from the raw data at left, informing maps of human cell architecture. And then those maps, as different machine learning developers request, you can either use the raw data directly or you can use the derived maps in these advanced machine learning systems. So what are those data types and what are the cell models in which we're generating those data types? We identified two primary models. Human-induced pluripotent stem cells as sort of the mother of all human cell types, as well as this breast cancer cell model and also an osteosarcoma cell model. For the cancer cell types, we look at them in the naive, untreated state, as well as under two chemotherapies. And then in the iPSC cells, we look at them again in the naive, undifferentiated state, as well as in neural and cardiac lineages. So you can think about those as two triangles with 6 points of each of those triangles, and those are our model systems. And for each of those points, we now interrogate the cell population with each of three fundamental modalities. One is we map protein subcellular locations with immunofluorescence microscopy. Two, we use proteomics with mass spectrometry to map protein-protein interactions, or PPIs, and their higher order protein assemblies and complexes. And three, this is a functional measurement, we knock down each of the genes in those cells, or at least large numbers of genes in those cells, and measure the effect on the transcriptome, so-called perturb-seq. Where are we right now in our progress? Here again, the two models are shown as the above and the below. So our largest progress in terms of counting genes and proteins, you'll see it depends how you count and what you count. But if you count genes and proteins, we're farthest along in the iPSC model, where we've mapped a genome-wide perturb-seq experiment, and that was reported last year. We've also now mapped a complete proteomic measurement of all, essentially most expressed proteins in that cell type and their interactions, and also gotten pretty good coverage with immunofluorescence. As you'll see, per protein, an image is a lot more work. Okay, so we'll talk about that in a second, but this is pretty good coverage of that model cell type. And then shown below, we also have a complete mass spec profile of proteins in the triple negative breast cancer cell model, as well as in the osteosarcoma model, and hundreds of proteins mapped for the other modalities. And I'd like to just mention that all of this data now is in excess of what was announced or planned in the original proposal. So originally we had really been more targeted, and given the sort of economies of scale we experienced about 2 years in, we renegotiated with program, not to change the budget, but to given the budget we had, we thought we could get more coverage. So I mentioned that if you count proteins, that's one story. If you count images, and data points and data volume, you'll see that's where the immunofluorescence images really, it reveals how much data there really are back there. So it's really a ton of data, we think, for these 2-cell models. And again, we still have the rest of this year in which new data releases are planned. Now, in terms of derived data types and tools that we've rolled out, of course, you aim to arm the AI machine learning community writ large with these data. But where we are with our own efforts is as follows. So first thing you can do is you can build a series of foundation models, one for each modality, and then as well as an integrated model, which integrates the 3. And in that foundational space, you can look for clusters of genes and proteins as evidenced by all 3 modalities. So what I'm showing here is a hierarchy of about 275 objects inside of cells. This is the osteosarcoma model that one can detect when you begin to integrate those modalities. So a circle or a node here represents a group of proteins with similar images, similar interaction patterns, and so on. Alternatively, to a hierarchy or to this sort of top-down hierarchy, you can view the same hierarchy as a bunch of nested circles or containers, because in a sense, that's what cells are, is they're containers, at least at a sort of writ large, they're containers for proteins nested inside one another. And both of those visualizations are facilitated by Cytoscape Web. Now, that's mapping the objects inside of cells. Given those foundational maps of cell biology, Emma Lundberg's group, and she and her postdoc Jan are here, and we'll talk about this later, are exploring, can I now use that knowledge to generate maps of cells or forward simulate what each of those data modalities would look like if I did the experiment in either the cell type under interrogation or a new cell type that has not yet been interrogated, but again, for which they can show they can achieve accurate simulations. So this is Protocelli. They have a preprint online. Please do check it out. And the idea is they can now, you can choose a cell type of interest, and they can show you what the microscopy image, that is to say the subcellular localization of that protein, would look like if you were to do the experiments. You can think now about using this foundational knowledge, data, and maps as a way of creating virtual cells. I'm sorry, I somehow don't have permission to show the video. Do I dare? No. Anyway, there's a nice video of doing that, but I'm sure Jan has a talk about this later in the lightning session. Just one more slide of statistics to show before I close. You can look at the number. So we have a robust cm4ai.org website now that's been live about 18 months. So you can track the number of visitations to that. You can track, of course, tools and data downloads. It's not as straightforward, however, to sort of distinguish those. Like I mentioned, a lot of what these tools downloads are really doing is providing access to derived data types. So so I would sort of add those together. And then, of course, you can look at how many people are viewing in terms of the most derived maps we now have online, viewing these cell maps in Cytoscape, as well as using our instructional videos and so on. We are proud of our citation count thus far with the papers we've published. This is all really in the past 12 to 18 months. Keep that in mind. These are all new projects. A couple of last points at the bottom here. Our team was selected for a presentation at the upcoming BioIT Hackathon next month. We're very excited about that. We'll talk a lot more about these data and its uses. We are currently hard at work in a Bridge2AI wide sort of way with Kyle and others in planning a dream challenge around our data. And the idea is you'll use the existing data plus other public knowledge to predict new data that are not yet revealed and will be held back, used for the Dream Challenge. And that is now, funding for that has now been provided, and that planning is underway. Thanks very much to Shurjo Shen and others for facilitating that. And then, of course, we're going to continue our Code Fest, and there's a Code Fest planned for the early fall. Please do take interest. I guess you're in the room, so I'd invite you to the lightning pitches, but you'll be here. Please, if you liked what you heard, please do give your full attention to Jan, Yulia, Sungjun, and Zhao Yu as they take you through various aspects. We also have 6 posters. We've tried, I think, very hard here and with planning to sort of expose all aspects of the project, from teaming down here and people. Jandos is going to talk about his knowledge graph, to ethics, like Julia will talk about, to standards from Tim Clark and others, in addition to the raw data types and the tools. So with that, thank you very much.
Thank you. That's very, very exciting. So now we're going to move on from the data generation projects to our working groups. So go.
And while Monty is loading that.
You need a microphone.
Working groups have 5 minutes to present. After 4 minutes, I will stand up and I will keep standing. And if you're not done after 5 minutes, I'll start dancing. And I'm not sure the room wants to see those antics. So please try to stay on time, but we're really excited to hear your updates.
And with that, level leader.
All right. Hello, everybody. So first up on Bridge Center talks, I'm going to talk to you a little bit about the standards working group. So to refresh your memory, in case you haven't been in contact with us, and I hope you have, but our initial goal was to make sure that the standards used across all of our wonderful GCs were, first of all, generalizable across all of our potential applications, especially beyond Bridge2AI itself. That we were able to produce reproducible and credible outputs, and we'll define those a little bit shortly as well, for the broader community, that they're updated efficiently, often, and consistently, and with clear provenance showing, what actually happened in that update, that there's transparent benchmarking and reliable, clearly defined quality assessment, and finally, that we're able to support responsible, trustworthy, and well-documented processes for AI readiness. So is that what we actually did? I hope I can convince you that we did at least some of that, because just as a short list of our accomplishments, we have in fact established some reusable standards for metadata capture that can be used beyond Bridge2AI, but work especially well for the data that's been generated here. That includes things like the Fairscape platform, which works especially well with the RO crates, which you heard Yael mention earlier. So we've deployed RO crates and systems for consistently identifying the metadata that should go within this packaging system across all of our GCs. We've also expanded the data sheets for data sets, which there's a poster about, you can hear more about as well, but those go into even further detail about defining the metadata that you'll find for any of our GC's data releases. We've defined AI readiness in specific metrics across varying different data types, so not just the research level, but image data, genomic data, and so forth. We built an entire Standards Explorer platform for literally exploring the landscape of biomedical standards, both within Bridge2AI and beyond. We also have an individual project where we've connected Bridge2AI with the GA4GH standards and applied it to the VRS standard, that's variants with a local rare disease cohort. We've been able to look into that further, and that's leading to efforts where we can actually use the phenopackage standard across Bridge2AI data wherever possible. So the Standards Explorer that I mentioned, and you can go ahead and check that out yourself, that is live in its beta release. There's a poster on that, or at least there will be a poster up for the session. But yeah, we think this is a really great resource, ongoing development, but fantastic for learning more about the kind of standards that might be applicable to your own data, but in turn, the standards that are out there that can be reused are in ongoing developments. And we've taken great pains to identify the areas where these standards are in active application for AI and ML uses in biomedicine. We've also established some, I don't think it's too much to say that these are kind of landmark manuscripts in the field. Kind of the top one being this AI readiness criteria for biomedical data. So hopefully you'll be able to see that in scientific data quite soon, but you can read it for now on in preprint on bioRxiv. So that's feel free to cite that anytime you need to know about what makes data AI ready. This is a question that keeps coming up in Bridge2AI and honestly even well beyond biomedical research. But we have some metrics in there specifically defining that, what goes into making data AI ready. But I alluded to the fact that we've also been looking into more specific data types. So that includes medical imaging data, genomic data, and metadata. And we have manuscripts out for each of those. You can read those in preprint. We also have a few more in the pipeline about things like those more specific data standards that I mentioned. And yeah, with that, I'll wrap up and lead on to the next working group.
Thank you, Harry. Communications, Lucila.
Thank you. I'll avoid any dances or anything. So we'll be quick. So in the last 6 months, we had a virtual meeting led by Aaron, Carol, and Pepe. Was highly successful with great response, especially for the fireside chat session. We've been active in LinkedIn and Blue Sky and have been conducting the colloquia with diving deep into breaking research or addressing topics of general interest. We revamped the portal and we provided more info on data sets. Again, the desire to have a common portal or a common focus so that we could branch out and link to the individual DGPs has been done. The colloquium series, in terms of, these are just a few examples, and in the theme of promoting the junior and emerging leaders, here are some examples from single cell analysis to AI for rural healthcare and explanation of the Common Fund data ecosystem. We also enable data discovery with the Bridge2AI website, in which again it points to the data sets that the DGPs have created, some data tools and products, and best practices. So that's all in there. In terms of LinkedIn, we have several thousand impressions. In here are some examples, again, featuring the emerging leaders in terms of events, in terms of posts, and some statistics on this is example of science in broadcast. So here, shout out, these are just 3 of them that selected randomly. Shannon, Alex, and Jake posting on what's happening at these meetings. Hopefully some will post from this meeting as well. In terms of, we have seminar recordings, they're all there for you to look at. There are educational shorts and community posts as well. So please take advantage of that and get familiar with the several resources. They're there beyond the data sources that we have heard about. In terms of building a community for science communications, again, publications, we are tracking those external perspectives in science conversation. And I'll talk a little bit more about publications soon. In fact, here, blockchain. Oh, sorry. Sorry if you couldn't hear. So Bridge2AI blockchain update, we have developed a prototype together with AI Ready in order to keep track of usage, to keep track of provenance of data, how data transformations happen, the code, related to pre-processing and all the way towards utilization and publications derived from it. So from publications, we can, consult do APIs with PubMed Central if we want to, in terms of who is using or who is declaring use of Bridge2AI. We could map those also that have requested data generated publications but have not cited the sources, which would be a problem. So the whole thing of generating this data sharing index, for example, which is of great interest to NIH, is embedded in such a prototype that would track utilization, that could embed smart contracts in it to have the data use agreements in it, in the release of data accordingly. So again, the prototype has been developed. There is submitted a demo for the AMIAS annual symposium together with AI Ready colleagues, and the framework is extensible to any other data set. So again, talking about reproducibility of results, how can you do that if you don't use the exact same data, the exact same data transformation, the exact same outlier removal than the original papers? So anyone here who tried to reproduce papers know it takes a while, it takes more than one postdoc sometimes, just to reproduce and then to do the next step. And all of these are related and should be acknowledged. That there is one point about recording of this, so that credit attribution could come not only when publications do cite the sources, but naturally, as if this data set was used and was processed in certain ways, the creator of the pre-processing code should be acknowledged. The original data source should be acknowledged in a whole a series of other things. So that can be automated. I mean, it does require a little bit of change in mindset. We heard from Yael that, there can be several data requests. Some can be spam. So how do we ascertain sources and then the products? Because just accessing the data is not enough, right? Someone must do something with it and generate a scientific product. So today and tomorrow, you will hear from Moni and Yulia about many things. So thanks for emphasizing the emerging leaders and the new generation, because that's very refreshing. There's a great team effort by other session organizers and volunteers. And again, very pleased to see recognition of aim ahead in consortium scholars. Please participate actively, ask questions, leverage the face-to-face time, and dive deep into posters, which, as you may have already noticed, are quite impressive already. Some of them are yet to come. And support and offstage support to the lightning pitches as well. So in the coming months, plan the Bridge2AI meeting and open house in the fall, which we share by Alex, Yael, and Ezra. Alex, in terms of the open house, and Yael and Ezra on the main meeting. Mark your calendars. It is done. It's it's October 20 and 21st at the Hilton Rockville. So nearby, very convenient by the train station location. We need some more people for the colloquia. And again, please encourage your emerging leaders to come forward. It's a national meeting. Those of us doing promotions know how it's important to be nationally and internationally recognized. So please do that. And let Brianna know of your work so we can broadcast it to our channels. Thank you.
Thank you, Lucila. Goldie?
Hi, everyone. My name's Goldie Komaie. I'm from the University of Colorado, Denver, and I'm presenting on behalf of the Teaming and Collaboration Working Group. This is our team led by Dr. Moni Munoz-Torres, along with the leads from the Grand Challenges, including Dr. Yulia Levitis-Strekalova from Chorus, Dr. Sarah Singer from AI Ready, and Dr. Jake Chen. I'd like to ask if the people from our working group could stand so we could recognize you. I see Jake and Sarah and Grace, I think, is here. Oh, there she is. Oh, Colleen, yes, thank you so much. Our working group meets regularly on a monthly basis, and I just want to present a few of our highlights from the last few months today. In February, members of our working group facilitated a half-day workshop at the University of Colorado Anschutz Medical Campus. The workshop was an interactive, hands-on exploration of what makes large-scale scientific teaming successful. We had a mix of graduate students, postdoc students, research associates, senior instructors in attendance, and the participants represented computational biosciences, immunology and microbiology, biochemistry, biomedical informatics, so a variety of disciplines. Our workshop materials are available online via the QR code link on the slide, as well as in a poster next door at the Evergreen poster gallery. And we also have a manuscript in development describing the workshop content and the workshop evaluation results. All right, some more highlights of our recent work include 2 manuscripts in preparation. We have one titled Dynamic Teaming Framework for Large-Scale Collaborative Research. We also have another manuscript titled Building Social Infrastructure for Big Team Science. We wanted to also highlight another publication from Dr. Levitis-Strekalova and the Clinical Chorus Project team that was on building AI competence in the healthcare workforce, and that has already come out in the Journal of Clinical and Translational Sciences. All right. We have multiple presentations that are taking place this spring that we're excited about. 2 of these will be taking place at the Science of Team Science Conference in May in British Columbia. We have one poster called Assessment to Action, a practical team science implementation workshop. And then we also have a talk that will be there called Beyond Big Team Science, a dynamic teaming framework for large-scale collaborative research. We were also invited to a panel at the AMIA Amplify Informatics Conference in May in Denver, Colorado. And we're also proposing to give our Bridge2AI team science training workshop at the AMIA annual symposium in the fall. And then, as I mentioned, 2 of our posters are here next door. You can view them at this meeting today or tomorrow in the poster gallery. So please do so. We'd love that. And then finally, the teaming evaluation team, we've completed several different team-specific reports for the different grand challenges. We've produced an executive summary and a written report based on data from the consortium-wide teaming survey. And then we are also tracking the productivity of the Bridge2AI consortium via Google Scholar page. So that's publicly available. It's updated bi-weekly, and it includes peer-reviewed scientific publications and preprints. So that is our update. Thank you so much.
We have 2 more updates from our working groups. I believe this is, 3 more updates, right? Yes. It's you, Alex. Yes, no worry. It was true. I was just making sure that I was not fibbing.
Okay, morning, everyone. Okay, so I'm going to be really brief because everyone's been saying it's been 6 months since we met, but it's only been about 4 months. We met in December. I was like, did I lose 2 months? Okay. I probably could have lost 2 months. Okay, so in terms of the data sharing, dissemination, and the open house working group, just some really brief updates. We've actually spent pretty much the last few months working on the RFI that was requested by NIH, and there was a lot of discussion going on within this working group as to what types of responses we should have to NIH. Obviously, they were requesting for a lot of information on things that would affect the data sets as they're being released from the data generation project. So multiple sessions on discussion around the science and the policy of the new draft that was put out there. And this resulted really in preparation of 3 different responses from the Bridge2AI group, one of which was a joint response between AI Ready and Voice around biometrics, particularly, again, as you might think, questions about how voice data is being released, how retinal images are being released. There were lots of questions about this, and these were issues that were raised in the actual RFI. So responses were generated for that. There was also a joint response on the CADRs that were sort of being discussed. And so again, a lot of discussion was going on in the working groups with respect to what the implications would be for releasing the Bridge2AI data sets, particularly as some of the data was consented, some of it's not consented. Erich brought that up earlier this morning when he was presenting on behalf of Chorus. So many weeks of discussion. And all of this, like I said, the responses were sent in March. There were additional responses also relating to the overall consideration of bulk biomedical data, right? The quantity of data that is actually being released. The numbers were being sort of put out there as to what constitutes a threshold for data release. And so there was discussion about that. So in terms of next steps with respect to this, the voice team is actually going to be preparing a JAMA paper based on the perception from patients of government-owned CADRs, right? And how that is going to work out too. Okay, secondly, in terms of discussions in the working group, this was also brought up, and this was in the New York Times. Grace, you brought this up to us, and there was a lot of internal discussion about the article that was released, or that was published in the New York Times about the ABCD, or ABC study? No, ABCD study. At any rate, we discussed this article quite closely. We met with, and this was sort of a good discussion in conjunction with the ethics working group. And they had met with the ABCD PI, who knew a little bit more about the information that was, or the issues related to this particular New York Times article. And they really discussed the lessons learned, if any, to avoid a similar situation with Bridge2AI data. So again, a lot of good discussion sort of came out of this work. Okay, in terms of dissemination events, we submitted a follow-up panel session for the AMIA Fall 2026 Symposium. This will be held in November. We're waiting a decision on that, but the intent of this panel is to provide updates for the DGPs and further highlighting the data sets and lessons learned from phase 1 of Bridge2AI. And as Lucila mentioned, we are now beginning to plan for the second day of the open house, which is being planned for October 2026. We will now we're generating some things for ads. We're going to actually put this out. We're going to disseminate slides for everyone at NIH to share with their colleagues so they can actually save the date. The tagline is a work in progress, so you guys can complain about it if you want. But at any rate, we will be providing this to everyone so they can actually get the word out about the open house. The open house is intended to be for everyone. So it's not just limited to NIH. We want people from industry, we want people from other sort of community partners and things like that to actually be coming to the open house to celebrate what we've all done in phase 1. So I will stop there. Thanks.
Thank you, Alex. I think next up is Camille with our ethics. Yes.
Good morning, everyone. Can you hear me okay? So I want to give a little bit of history about instead of giving an update for the ethics working group and the things that we've done since our last update, I want to focus on something that we've worked on for several years. And it started with the branding of Bridge2AI being ethically sourced, trustworthy, flagship. And I'm not a legal scholar, but I get into the minutia of words, and I really wanted to know what these words meant. So in a meeting probably in early 2021, I honestly don't know what year it was, with Joe, with Lene, with several of us talking about data, I asked the question, what does it mean to be ethically sourced? And Lene said, that's for you guys to help us define. And I took a deep breath and thought, this is going to be really interesting and I want to tackle this. And with our LC working group, with people from our grand challenges over almost a year, we worked through and where I found and I've talked about this at several different points, is the term ethically sourced started with supply chain management. So I learned about supply chain management. We published a paper in November that involved many people from the consortium across the DGPs, different working groups were involved. And we published this paper in JAMIA Open about what that meant. And in probably December, January of this year, I wanted it to go further. I wanted it not to just be a paper that somebody would someday look at by chance. I wanted it to be a tool that we could actually use to help us think through what is involved with whether we are, or as Grace likes to ask, what would I tell my future grantees that they need to be thinking about when they're going to build a health data repository? So I've been talking over the past few months. I gave a presentation at the Data Sharing and Dissemination meeting April 1st, where I introduced this checklist. And I've received some amazing feedback, because the only way we can know whether or not this checklist tool is actually going to help any of us is by asking everyone who's involved with one of the grand challenges, how do we do this? What's working? What's not working? What gives you hope? What brings you angst? And this is where we are right now. We're in this process of somewhat discomfort. So one of the things that I learned from Barbara Evans, talking about Chorus and how this fits with Chorus, and as Erich was mentioning, they collect clinically sourced data that is patient data that has a whole different paradigm. And she said, and this has come up in talking with Tim as well, is that this term sourced, ethically sourced, brings people back to the very beginning of something. Where did it start? And this isn't about where did it start. It's whether in terms of supply chain management, does it ethically sourced means that it hits 7 different steps in a cycle. It's not just what happened at the beginning and did we get informed consent or did we have to? It's once do we need to justify why we need these data? What's the purpose of these data? How should we get them? Who grants permission? Once we get them, where do we put them? Who manages this? Who is stewarding this process? And I want to see her dance, so I'm going to keep going. And so one of the things I learned is that we're going to need to change the name. And so one of the new considerations is, do we change it from ethically sourced to ethical data governance checklist for AI-ready health data research repositories? Because this really is about governance. And so that was one of the first things that I learned from talking with Barbara, is that the name is tripping people up. So I started looking at CM4AI. They gave us wonderful feedback yesterday. And what we're having trouble with CM4AI is that several sections of the checklist don't matter to them because they're not using data that would be considered human subjects data. Legacy cell lines predate the current consent frameworks. The framework intentionally permits secondary use of cell lines without additional consent. So with respect to the checklist, I need to not just fix CD&E, because CD&E still works for prospective data collection. I need to create a new module that addresses non-human research data. Voice and AI ready are very much aligned. They're prospective, purpose-driven. There are certain characteristics that the checklist challenges with the voice in terms of re-identification. The teams need to document what withdrawal actually does and does not guarantee. And the DUA needs to address cloning, synthesis, and re-identification attempts. And with AI Ready, they've done some amazing work over the past years. They've employed a tiered access for their multimodal data. Retinal imaging is under safe harbor. Genomics is under controlled access. The Swiss cheese model that we've talked about over the course of years, which is layered. We use this at UC San Diego for our COVID prevention. They just published in Lancet, I don't know what eClinical medicine means. I think it's, I don't know what the E is. Anyway, so open question for all of you is whether the data security protocols for compliance and international data sharing are actually being addressed. How are we addressing those? For the other thing is that I work across several of the working groups. I've had the honor and privilege of working with Moni and team on the standards working group over the past year plus. I think it's been about probably 18 months. And they have the D for D, data sheets for data sets. I saw that we had, oh good, I get some dancing going on. I saw that we had some overlap, so I wanted to make sure we didn't. And now we know what we need to do. We have 6 changes that I'm proposing. I'm going to share these with you so you all can look at them later. But these are 6 changes that we need to do now to make this checklist more relevant. And for questions for all of you here, the title, do we endorse that? Do we endorse the 6 changes? How are the grand challenges addressing the DSP awareness? And what barriers and facilitators have you observed in your work with this checklist? And with that, I will stop. Thank you.
Thank you very much. And last but not least, Jack Rincon. And are there slides here or no? Okay. Are you on Zoom or?
I'm not. At the initial.
Yes. Christian may be able to facilitate if you need to share any materials.
Yeah. Can I just have you go to the website?
Sure.
Is that okay?
Sure.
Yep. Okay.
Oh, interesting. Okay, we're doing this manually.
Thank you for your patience, you guys.
Pray that I did this right.
Okay. Thank you. All right, so I'm actually not. Can you all hear me? Hi. Thank you. I'm not going to really talk about the portal. Instead, I'm just going to say a few words, if that's okay with you guys. Someone. So it is truly inspiring to see so many brilliant trainees and learners gathered here today. Each of you representing the next frontier of medicine. However, this would not be a meeting that fully appreciates our progress if we didn't hold space and time to acknowledge the faces we have inspired and the colleagues we have collaborated with to invest in people. So to illustrate the profound impact of the Bridge2AI learning resources, I want to highlight 2 individuals who exemplify the kind of growth and sustainability this program fosters. First, we have Stephen Swee, whose journey from chemical engineering, I think he's here with us. Everybody, y'all can see him, whose journey from chemical engineering to a Ph.D. in medical informatics showcases a remarkable evolution in technology and technical complexity. Stephen has moved beyond traditional molecular simulations to pioneer a multimodal knowledge distillation framework that aligns clinical text with ECG signals to predict cardiac risk. His recent 2025 work predicting cardiac death from ECG-based multimodal knowledge distillation via variational inference highlights his ability to turn diverse data into life-saving insights. Then there's Erich Adam. These gentlemen's over there. He's here with us today. An AI research scientist whose work is a masterclass in explainability and multi-omics integration. Erich has developed sophisticated graph LLM frameworks that integrate molecular interaction data with clinical notes, ensuring that AI reasoning remains grounded and transparent for clinicians. His 2025 publication, Athena, Atherosclerosis through Hierarchical Explainable AI for Personalized Risk Stratification, exemplifies his leadership in using hierarchical neural networks to provide clear, actionable medical insights. The success of Stephen and Erich proves that the true sustainability of Bridge2AI isn't just in the tools we build, but in the people we invest. These trainees represent the incredible potential and the amazing contributions that will define the future of healthcare. Now, while I have a captive audience, I would like us all to appreciate the slide you guys have been staring at for the last minute or so, and just enjoy and appreciate these faces in silence since I have you guys stuck here for a few minutes. It's really all I have to say.
It is fast. Thank you, Jack, and thank you, everybody. So the price we pay for going over is that we might have a little bit less in the department of questions than we had hoped. But we have 15 minutes with us, and I would like to welcome anyone on the floor to walk up to one of the mics and share questions. These are the people you just heard, and Brad is ready to get us started. Please.
Okay, so for the DGPs mainly, so I'm curious about, so in the last 5 days or so, UK Biobank had a major, I'd say, snafu in that their data was being sold on Alibaba, and that was, taken offline after it was discovered. And UK Biobank their enclave is now shut down for about a month while they're trying to figure out what went wrong. And so I'm wondering if the DGPs have had any conversations with their general counsels or with their organizations more broadly to just try to figure out how that is going to affect your data sharing initiatives.
Thank you, Brad. Who would like to take the mic? Yael, you seem eager.
Yeah, I mean, it's a little, thanks, Brad. It's a little early to talk about it. I did meet with my general counsel right after. I think it's interesting I'm going to be careful not to say things. It's it's really close to cybersecurity risk, right? So I also have a role in my hospital, and hospital health systems have looked into this, right? It's not just data sharing. We have to learn from hospital systems that are treating this as cybersecurity risk, as our biomedical data is being extracted from our hospitals and shared for research. So I think we started having conversation, you and I, through emails. But I think that's where we have to go, right? As researchers, we had a little bit of data. Hospitals had lots of data. They had a lot of work in cybersecurity. We had little. And now as we're extracting all that data to the research side, I think that's where we need to communicate more with our hospital systems that have gone through this extensively. For example, at Sick Kids in Toronto and pediatric hospitals, I think 2 years ago, they had a major cybersecurity breach, everything shut down. So we're not going to, I don't think we should reinvent the wheel. I think we need to learn from hospitals. I obviously don't have a solution yet. Yeah.
Thank you. Erich.
Yeah, I talked earlier about HIPAA covered data, and I would just say that I caught most of your talk, coming on in the hallway. So the challenge is that if you were to ask each of our 14 centers for a HIPAA waiver, which would allow you to share the data with a non-HIPAA covered entity, they would have to know that there was so-called minimal risk. And the minimal risk has to be defined by each episode of sharing. So it's not like it's minimal risk because in general we're doing X. It's minimal risk because this person's using it in this way. And they're also, adhering to the HIPAA principles. So the downstream risks are the challenging ones. And so from our hospital's general counsel, I think their concern is that, UK Biobank would, let's say that were us, would we be sued for that or held criminally liable when we thought we gave it to someone who signed a DUA and did everything right? And there is significant concern that we would be liable or at criminal risk from not vetting that individual. Similarly, the NIH does not yet have a policy on how to handle the DOJ rules about sharing with countries of concern. Really, that's only for entities, but it's not necessarily, NIH does not define it yet for the individuals themselves. And so, you can share it within the US, but if if you're sharing with an individual who belongs to another country, that's at risk. So very fraught and not yet clarified, I think, even by the federal government.
Trey.
Yeah, so Brad, in terms of the UK Biobank, we work a lot with like Naomi Allen, who's chief scientist at the biobank over there, and I think she was quoted in the Guardian as saying she was terribly cross about the situation. They're underwater right now, dealing with, as you can imagine, everything. Anyway, I think they're doing the forensics on what happened. If I had to take a wild guess, again, we'll see what they uncover. Years ago, they used to give out the UK Biobank to researchers to host internally, and they stopped doing that. Maybe this is a good reason why. It'll be interesting to see whether it was data taken out of DNA Nexus, where the data, which is now the trusted research environment, that's the TRE that hosts the UK Biobank. It'll be interesting to see whether someone got it out of there or whether it's just a archival copy that was never deleted properly from years ago. But anyway, I think in terms of this project, hopefully we're planning things with all that wisdom behind us and properly into the future. That said, I think just one last point. What I really worry about with that news is, if you look, like, members of parliament over there are aghast at the $1 billion+ that have been spent on biobanks and just to, expose their people to privacy concerns, and why would they want to continue doing this? So I think I think the risk isn't just to the UK Biobank, but it's to the whole notion of longitudinal population health resources over there, which of course, as you know, has been a huge leader for all of us. But no, but anyway, no pun intended. Actually, that's debatable. But anyway, I worry about this sort of the bad press. I think we can all see and understand what happened, and they'll figure out exactly what the forensics are. But I really worry about the sort of effect it's going to have on the public and their perception of these things.
Thank you, Trey. Hello, this side of the room. Any questions for any of our researchers? Any additional answers to Brad's question? Yael?
I can just add, we talked a lot about the New York Times article with the ABCD, it was not a data breach, but the wrongful use. And I think what was really interesting is the answer letter from the ABCD PIs that we haven't shared enough, and explain how much broad data sharing ethically is actually helping patients and helping kids, and the balance between help and harm. And I think, obviously people like the New York Times will always amplify when there's a breach or when there's a problem, but not amplify how much data helps. I'll always remember my conversation, I think, with Trey and Tim the first year at CFDE, where you told me why don't we tell the patients of people who have cancer, the families of the patients, that we can't share biomedical data that's going to save them and they're going to die before biomedical data is being shared for ethical reasons. Tell them that we can share data ethically. That resonated, and I understand it better now.
Thank you. That letter is shared on the Slack all hands, by the way, if you want to read it. Camille.
Yeah, thank you. And with regard to ABCD, the person that presented at the ethics working group was Pilar Osorio. She's one of our external board members or steering committee members. And she's on HBCD, just for clarification. But what this is bringing up is people like Barbara Evans and Pilar Osorio. They are experts. They're law professors. They understand the nuance, and the regulations are complex. And I know that when you talk to your institution's legal counsel, you're hoping for the best, but this is a very niche area that's very new and fraught and evolving. And I think what you're just raising, Yael, is such a critically important piece of telling the story of what's going well and keeping some form of, well, actually developing a form of bidirectional communications with not only people who are contributing to these data sets. Don't just leave them at the very beginning and go on and on. That is critically important for public trust, and we know how to do it, and we need to be doing it. And one thing for the sponsors is when sponsors mandate a requirement for communicating results beyond PubMed, but back to the communities that are contributing in a way that's accessible to these communities, that can really not only build trust, but it can change the game for future biomedical research. So I'll leave you there. Thank you.
Thank you, Camille. Do we have, yes, Joe.
Sorry, I was going to stay quiet during this whole meeting, but
Down to the.
that last conversation, I couldn't stay quiet. Yeah, of course, there's benefits to doing this research for people, but the problem of all of this stuff, precision medicine or AI, is that it purports to help everybody, but because the research side is egalitarian and altruistic and collects massive amounts of data, the development side, the people who actually get helped, is based on a standard curve. And it's even skewed a little bit to the right. It's people who can pay, right? Who people have insurance, people who know how to mitigate the system. So one side is a flattened curve, if you will, so more people get help. The other side is a very steep curve where only some people get help. And in this system, the same is true for harm. In the ABCD study, it was African Americans that were harmed. In the genomics area, it's going to be rare populations, people with rare diseases, people that come from island communities, Native Americans. They're going to get harmed because of the way that products and health systems are delivered. So I just wanted to say that.
Joe, that's very insightful. Do we have other questions? Okay, we're quiet. Oh, come on, Cecilia.
Yeah, I was gonna mention, I think, so worrying about the access and then also the benefits, we're talking about equity, because even like last night when AI Ready group met, we were concerned that, people would have to pay like $300 to be able to download the data, because, a lot of people may not be able to afford it. And then, but then who's going to pay for the TRE for everyone, in the world, what kind of modeling are they going to do? There's no right answer. But then also kind of Camille's perspective in terms of like balancing, and then also addressing, also kind of engaging with the community or the participants ongoing, and then kind of highlighting both the bad actors, but then also the good outcomes, or what we've been able to achieve. I think it's a very complex issue in terms of access, and then who benefits from it ultimately. But I am grateful to be part of Bridge2AI, because I do think that we are leading a lot of discussions, and then we've definitely charted our own territory, because this is a new world.
Thank you. Thank you for raising that point. This is definitely uncharted territory. And part of that spirit of the experiment at Bridge2AI has been from design from the beginning. Thank you. Okay, with that, I think I'm going to give us a 7th inning stretch because we have come to the end of our reports. So I want everybody to please give a huge round of applause to our speakers. And now we're going to transition. So we have been listening to our reports within Bridge2AI. We just heard our grand challenges in our working groups, but we're now